right. I think we should get started. So yeah, thank you all for coming. And it's a real great pleasure to introduce our VTLSS speaker for today, Dr. Natalia Dudareva. She is a distinguished professor in the Department of Biochemistry at Purdue University. And I wanted to give you a little bit of background about Natalia. She did her first PhD studies in biochemistry and molecular biology at the Institute of Biochemistry in Kyiv in the Ukraine. And then she came to Western Europe and she got a second PhD with honors in plant molecular biology from the University Louis Pasteur in Strasbourg in France. Then through Canada, she came to the US, where she did postdoctoral studies with Dr. Iran Pichersky at the University of Michigan. And that's really where she got first exposed to plant volatiles and plant volatile biochemistry and molecular biology. Then she went to Purdue. She became a professor there, first in the Department of Horticulture and Landscape Architecture. And since 2013, I think, right, she is distinguished professor in the Department of Biochemistry. And she's also, since 2020, the director of the Center for Plant Biology in the College of Agriculture at Purdue. Natalia and I, we go way back. I think it's more than 20 years ago that we first got to know each other while I was still a postdoc at the Max Planck Institute for Chemical Ecology in Jena, Germany. And she did a sabbatical in 2003 there. And we worked on one of the central enzymes in terpene volatile metabolism in Snapdragon. And so Natalia tries to address with her research always fundamental mechanisms, whether it's in metabolism, metabolic flux, metabolic engineering of plant volatiles, whether it's with subcellular regulation or circadian rhythms, or as she will show us today about emission of volatiles and perception of volatiles by plants. And she never really rests on a discovery. She always pushes boundaries. She always asks the next interesting and challenging question. And so it's not really surprising that her group has published or is publishing their findings pretty much regularly in the highest profile journals. I couldn't really count, but she just said it's at least 150 publications, which are all very highly cited. And she received, of course, many awards. I just want to highlight a few. She's an elected AAAS fellow. She has served as chair for the Gordon Conference of Plant Metabolic Engineering. In 2016, she received a prestigious Alexander von Humboldt Research Award in Germany. And since last year, she was named an ASPB fellow. ASPB stands for American Society of Plant Biologists. And just this year, she has been named a pioneer of the ASPB for her contributions to the plant biology community. So today, she will talk about her latest discovery on how plants release scent and volatiles and how plants actually can perceive these volatiles, which is pretty cool if you're thinking about plant-plant communication. And with that, please help me in welcoming Natalia.
Thank you, Dora, for your nice introduction. Thank you for coming for my talk and for your kind invitation. But only what I want to say, I don't collect PhDs. It's just by by situation. It's happened because my first PhD was in animal field, and I started epigenetics in rats under hormonal treatment. And since I moved to plant field, I wanted to study and to prove that I can do research in plants. And today I'm going to talk about our latest discoveries in plant volatile field. But what I want to say is that we all lived in the field of volatile compounds. And today, volatiles are heavily used in cosmetic and fragrance industries, and biologically active compounds are used in aromatherapy. But volatiles were really very important for humans from ancient time. And already in 1300 BC, Egyptians were isolated primitive perfumes and used in their religious rituals. But the first actually alcohol-based perfume was isolated in 1370 in Europe, and it called Hungarian water because it was isolated in Hungary. And it was also called spirit of rosemary because it was isolated from rosemary. But 14th century became actually recognized for cultivation already flowers for their perfume essence. And it happens on south of France. And even right now, this is a major production of flowers for perfumes. However, it took practically 500 years before humans developed methods to detect volatiles. And in 1980s, it was developed a headspace analysis of plant volatiles. And the name headspace is coming from beer industry, where volatiles were first detected in a head of the beer. And practically next years are known for developing of detection profiles and sampling of volatiles. And GCM math became more sensitive, and people started to detect volatiles which present at very low levels. And by 1993, 1700 of volatile compounds have been already identified in from 90 plant families. It took only 3 years to isolate first gene, and I was really lucky because I was participating in isolation, and I was a first author on this paper which we published in Plant Cell, and the first gene responsible for volatile compounds was linalool synthase. And then in 2006, we published a book together with my postdoctoral advisor, Ran Pichersky, on biology of fluorescent, where we summarized at that time all data about molecular biology, biochemistry, function of volatiles, and evolution. By 2017, our first transporter involved in sand formation has been identified in my lab, and we published our paper in Science. Because before that, people saw that it's a simple diffusion of volatiles out of the cell. And in 2020, we published a second book, and not because we wanted to publish second book, because, you know, editors, they ran off of old books and people wanted to have a book. They contacted us and said, okay, could you put a second edition? And I did not want to do it, but Ran said, okay, let's do it. And we said, okay, we will name it biology of plant volatiles, and we expanded to vegetative tissue. But plants produce volatiles not for us, not for our pleasure. They play important roles in plant reproduction, defense, and communication. And they also contribute to stress protection, atmospheric chemistry, pest control, and biofuels. And volatiles emitted from flowers, they provide a chemical cues for insects and ensure a reproductive success of plants. And while plants can be, flowers could be very similar in color and morphology, they always produce different scent bouquet, which consists of volatile compounds, which is small molecules with low boiling point and high vapor pressure. And they present a different combination in scent bouquet. And in such way, there is no two flowers which have exactly the same scent due to relative abundances and interactions of these compounds in bouquet. Volatiles emitted from fruits, of course, they contribute to flavor of fruits, but also they contribute to attract seed disseminators. Volatiles emitted with vegetative tissue, they contribute to plant defense, and they can repel or intoxicate attacking herbivores, or they can also attract natural enemies of attacking herbivores. What is important is that emitting plants after pathogen attack could also inform neighboring plants about pathogen attack, which is called like priming, and we will come to this point a little bit later. And as I mentioned already, as I protect against stress, especially here, you can say no isopene can be used as a biofuels, a lot of terpenes or phenylethanol here. They can use for pest management, but what is important, they also contribute to atmospheric chemistry as they produce these organic aerosols, which use as a nuclei for condensation. And in such way, they will contribute to climate as well. If you look every single day, 20% of photosynthetically fixed carbon is released as waste compounds. It's really a very huge amount of energy which takes to fix carbon and, to release it again. And further increase in temperature by only 2-3 centigrade Celsius will increase global emission by additional 30% to 45%. What it means that considering that in past 30 years, we see emission increase only by 10%, here we see really a very huge increase in near future. But how it affects actually plants? And what is going, it will increase biosynthesis and volatility of compounds on one place, but it also will lead to actually changes in flowering time and fruit development. And in the early days, I wanted to see effect of temperature on snapdragon flowers. I put them in growth chamber and increasing temperature. They start to flowering much, much faster. But what it will lead to altering overlap between a flower phenology and insect availability for pollination. What it means that flowers will be ready for pollination, but it will be no insects there. And also drought stress might result in degrees of volatile compounds. On the other hand, elevated ozone level will lead to degradation of certain volatile compounds, and you can see here how they will decrease, degraded. And it will also reduce the distance distribution from emitted sources. And you can see that insects which are far away from plants, they will not sense these compounds due to decrease of distribution of them. And as a degraded compound, it will be new compounds which insects don't know, or it will lead to production of other inducible volatiles. As a result, it will increase pollinator searching time, reduce their foraging efficiency, and diminish actually pollination service. And overall, if you look on the effect of global climate changes on actually volatiles and their perception, it will also alter insect perception volatiles, could lead to disruption of plant defense and reproduction. It can have profound effect on plant-plant communication and an effect of climate warming by aerosol formation and direct or indirect greenhouse effects. This is why it's really very important to understand how plants produce volatile compounds, how they emit volatile compounds, and what we can do to prevent maybe unwanted emission of volatile compounds, or to direct it to a certain period of time when they're required for defense and communication. And here I did not mention, but practically you can't find any literature about allergy for volatile compounds, but I have been exposed to personally to high level of volatiles when we were working with it. Right away, you're getting tired and you're getting headache. In this case, high emission will not be actually very good for humans as well. If you look on emission of volatile compounds, emission practically depends on rate of biosynthesis and rate of release. And the rate of biosynthesis is species specific, especially developmentally and ionally regulated. And even close related species, which rely on different pollinators, like here, Clarkia cancina on bee and flies and Clarkia burai on moths, they produce different volatile compounds. And level of volatile compounds is also high when it's flowers are ready for pollination. And here you can see snapdragon flowers is from day 5 to day 8 with highest emission. And very often it changes rhythmically and coincides with activity of potential pollinators. And you can see here, snapdragon flowers, this is emission of methyl benzoate. Petunia and snapdragon produce methyl benzoate both. And but snapdragon produce methyl benzoate during the day when it's high activity of bumblebees, and petunia produce during night when it's the highest activity of moths pollinated. And also emission depends on physiological factors, which is pollination and pathogen infection. And practically plants don't shut down emission of volatile compounds right away after pollination, because they want to be ensure that fertilization took place. And here you can see that it takes 48 hours for snapdragon flowers to fertilize the egg, and for petunia 36 hours. After fertilization took place, plants shut down their formation of volatile compounds. If you look on okay, sorry, but it's looking like it's somehow a part of the slide. This is it. Why it's rate of release is not here. Okay, probably it was next slide. But what I want to say is that rate, I will go back a little bit, and rate of release here, it depends on physiological properties of membrane itself, which plants needs to cross, and physical chemical parameters of volatiles. And our big question was if any biological process are involved in release of volatile compounds. Within the sand bouquet, practically you have a mixture of volatile compounds which synthesize at different levels. And for comparative analysis of volatile emission, we invented like VSE emission factor, which is a rate of emission to rate of biosynthesis, emission flux to biosynthetic flux. And if VAF is equal to 1, it means that emission of volatile compounds is regulated by biosynthesis. It means that as much as biosynthesized, all is released. If VAF is less than 1, it means that there is some barrier or something which is keeping volatiles inside, and then whatever it's synthesized, not all released into the atmosphere. And if you look on different volatile compounds, you can see that volatiles with the highest volatility, they have VAF very close to 1. While volatiles with relatively low volatility, they have VAF less than 1. And for high these volatile compounds, you can see that biosynthetic flux and emission flux overlaps. This is what I'm talking about these methyl benzoate and benzaldehyde compounds. You see how nicely it's overlapped. But for relatively low volatile compounds, you do see this shift between biosynthesis and emission. You can see that something is really affecting it. Also, what you can see is that highly volatile compounds, they mainly emit it, and they have very small internal pools, while volatiles with low volatility, which is present here, they mainly present inside cells at low emission rate. Okay, for volatiles to be released, they need to cross, we're coming to this slide. They need to cross plasma membrane, cell wall, cuticle, and then they will emit to the atmosphere. And This is what I mentioned already. Rate of release depends on physical chemical parameters of the compound, volatility, diffusivity, octanol water partitioning coefficient, and various causes, membrane lipid composition, soluble porosity, and cuticle composition. And release will also depend on physiological factors, light intensity, temperature, and relative humidity. However, until now, it was not known if any other mechanisms are involved in the release. For a long time, it was accepted that volatiles simply diffused out of cells. And it was really very difficult to convince or change people's mind. And it took 3 times the submission of NSF grant. And before we did not have really already proof and calculations, nobody believed me on it. And what we wanted to do, considering this movement of volatiles, we decided to actually to make a calculation to prove that biologically active mechanisms are involved. Since we could detect internal pulse and emission, we decided to calculate how much we need to have inside cell to sustain emission rate, which we determine experimentally by diffusion itself. And we use this, fixed first law, where emission rate is proportional to concentration along the diffusion path and divided by resistance, where you have all these like properties of diffusion barriers involved here. And we decided at steady state balance state, you can see that flux at each barrier will be equal, and then biosynthetic flux will be equal to emission rate. And we made a calculation for 2 compounds. One was methyl benzoate, which is benzoate compound, and one is mirolidol. And our calculations showed that you have to have 120 millimolar in plasma membrane for methyl benzoate and 48 millimolar of mirolidol at plasma membrane for nerolidol. This is huge concentration. And even, there were data when we searched the literature that formula of linalool practically disrupt plasma membranes, and you can lead like a cellular content can leak out of the cell. This practically showed right away that biologically active mechanism must exist to lower barrier resistance, thereby reducing basic concentration on plasma membrane and likely concentration, internal concentration. This is why we started to look for possible mechanism involved in VSE movement, and we proposed that probably VSE could be taken in lipid droplets or in bilayer of vesicles to the plasma membrane. Across plasma membrane, it could be exocytosis or transported, and then probably through the cell wall, volatiles could be moved by lipid transfer protein or other carrier protein, and from cuticle you will have simple diffusion. Why do you need here some carriers? Because you want to prevent repetitioning of volatiles back to the plasma membrane. And what we did, we searched first for potential transporters involved. We generated RNA-seq data sets at the time of low emission and high emission, and we found one ABC transporter which was upregulated 100 times. And actually, we were lucky that already Rob Schurig group partially characterized this transporter. He did not know what this transporter was doing, but he showed that this transporter is regulated by ADARAN, a transcription factor, which regulates emission in petunia flowers and in localized to plasma membrane. 2 experiments which we did not need. But then we generated transgenic plants, and when we generated transgenic plants, and with non-regulation about 80 to 90%, reduction in RNA level of this ABCG1 transporter, total emission rate event went down and internal pulse increase. And we did experiments. It was very interesting. If you look through years of research, initially it was only biochemical approach and we could identify protein and prove activity, but it was no really transgenic approach to prove the function of a gene in planter. And each time when I would submit my first papers, people would say, oh, prove it in planter. Okay, now we took another approach. We prove it in planter. People say, no, please prove it biochemically. It looks like it's you need to have two approaches to be successful in publication. This is why we overexpress this transporter and divide to cells. And we did assays using radioactive metal benzene. And depending on orientation of transporter, you can expect higher radioactivity in your transgenic lines or lower radioactivity. In our case, it was lower radioactivity transporter was taking compound out of the cells. And what was interesting is that the transporter was specific for benzenate compounds, but it was not able to transport terpenoid compounds. And again, you know, when we isolated first gene, linalool synthase, Harold Baumeister contacted us and he said, could you give us linalool synthase? We will overexpress it in petunia. They overexpress it in petunia. They did see production of linalool, but it was never emitted. It was bound as a glucoside inside. And it was not clear why, because petunia does not have transporters for terpenoid compounds. Now it was crystal clear. But what is about toxicity? Our model predicted that all these volatiles at high concentration will lead to toxicity. We use two dyes with different action of mode of action. Propydium iodide first, which stains practically nucleic acid of cells with damage only if membranes are damaged. And this, you know, like circles inside, this is nuclei. It means that it's damaged membrane and it's getting in. The other one is fluorescent diacetate. Normally, it's unlabeled compound and it stains cell viability and membrane integrity. As a diacetate, it's getting in a cell, and esterase clips this diacetate moiety. Then it becomes fluorescent, but it can't leave the cell. And only it can leave if membrane is damaged. And you can see here, it's much less labeled inside. Now, the cuticle is the last step which volatiles need to cross. And it involves the process. And actually what we identify as a cuticle accumulates almost 52% of volatiles accumulate in the cuticle in normal cells. And partitioning through the cuticle is involved, partitioning from aqueous cell wall into the cuticle, and then diffusion through the cuticle, and then vaporization from the cuticle into the atmosphere. And what we decided, practically diffusion through the cuticle is a major barrier for VSE emission. And I want to give you one good example for young scientists that example when hypothesis was completely wrong. We hypothesized thinner cuticle will permit increased VSE release. And just remember, and this is how you need to see that not always your hypotheses are correct really. And what we decided when we identify ABCG transporter, we also identified a couple of transporters which were homologous to Arabidopsis transporters involved in wax trafficking. And it was known that if you disrupt this transporter, you can change cuticle composition or cuticle thickness in a plant. And what we decided to downregulate one of these transporters. It was ABCG12, and we got several, transgenic lines with downregulation, different levels of downregulation, and we decided to choose these 3 lines with the highest downregulation. And what we looked at, indeed, wax amount was reduced in these 3 lines, but wax composition did not change. And really, I think we were lucky just, that the only effect of this manipulation was the thickness of the cuticle. And thickness of cuticle was reduced by 22%. And you remember I said, thinner cuticle, high emission. This is first result. Emission was decreased. We said, okay, fine. Maybe something, But what was big surprise, that internal pulse were reduced. What it means that really biosynthetic flux, which is changes in emission plus changes, sum of changes in emission plus sum of changes in internal pulse over time was also reduced. And this we really could not really understand in the beginning. But when we look on VF, which was the ratio, it was again decreased. And well, it's interesting that you can see here that actually reduction in cuticle mainly affected low compounds with low volatility, but did not affect compounds with high volatility. When we look on the distribution of compounds, you can see that thickness of cuticle led to redistribution of volatiles, internal pools within the flowers. And they started to accumulate internally, and less it was in the cuticle because and you can see here that actually partitioning of VAC in the cuticle very nicely correlate with volatility of compounds. However, thickness of cuticle affect all compounds, and relative abundances of VAC, all VACs, and all of them started to accumulate within the cell. What happened? Most likely, you can see here the situation where in wild type you have 52% of volatiles in the cuticle. Now cuticle became thinner and less volatiles in the cuticle, but they built up inside. And this leads actually to toxicity, and you can see here that membrane become damaged, and plants sense it and feedback regulate actually expression of genes involve in formation of phenylalanine, which is precursor for all these compounds. It means that to prevent this toxicity, plants practically feedback regulate by synthesis of these compounds. And indeed, when we look for expression of all these genes, we found that they were downregulated and even transporter was downregulated. But what it means is a completely new function of cuticle here, because cuticle serves as an essential member of volatile metabolic network and works as a VAC concentrator. Now we look for LTPs. We identify LTPs out of 3 highly expressed LTPs. LTP3, downregulation of LTP3 led to decrease in volatile emission, although it did not affect any internal pools. But it led also for redistribution, and as soon as we block LTPs, more volatiles accumulate inside of the cell. Actually, facilitated VAC transport by LTPs here, it's analogous to oxygen transport by myoglobin in our muscles. And effect of LTPs on VAC flux depends on LTP concentration, on binding constant of LTP to VACs, and differences in concentration between the apical phase site and plasma membrane and inner side of the cuticle. And when we model it, you can see that always the flux will be higher in wild type, which can contain LTPs. And you can see it will be reduced when LTPs are downregulated. And if you calculate it without LTPs, just free diffusion, you can see that the flux will be reduced. And interestingly, that model predicted that flux will be reduced by 18%, but in general, what we saw in our RNA lines that flux was reduced by 49% relative to wild type, because practically model did not take in account that biosynthesis was reduced, and we could not model it in a model. Now, if you look overall on all these results, you can see that increase in barrier resistance always leads to decrease in VOC emission. However, alterations of subcellular barrier properties have different effect on internal pools. And here you can see in case of plasma membrane, internal pools increase. In terms of LTPs, it's no changes. And in terms of thickness of cuticle, it's reduced. Since emission from cuticle is simple diffusion, LTPs, this is small proteins which have very broad binding specificity, then the release of individual compounds practically determined by substrate specificity of AB sequence. Now, as I mentioned, that plants inform neighboring plants about pathogen attack. And actually, these interactions not only occur above ground, but also below ground. But and then plants are constantly targets of release compounds of plant communication, also plant insect and plant microbial interactions, because microbes also produce volatile compounds and plants exposed to it. And if you look, that it's like a two-way street. When plants perceive volatile compounds, these volatile compounds have to cross the same like cuticle, cell wall, plasma membrane, and then in cytosol they should induce signal transduction and cellular response. And practically nothing is known about these processes. The problem was with priming. Then when I talk even with people who work with priming, could you give me any idea what compound is specific for priming? They did not know. And prime plant does not have any changes. You can't detect any changes until it's not infected. But when it's infected, it's already too late because perception would occur. You know, it's like our immunization. We're immunized, we take a vaccine, but we don't see any effect before we are not infected, right? And several years ago, actually, we discovered interorgan aerial transport when we found that volatiles accumulate in different organs. We did actually profiling of terpenoid compounds. And when we look for a gene which is responsible for formation of these compounds, we found that it's actually TPS1, it's expressed in a tube. What it means is when at bud stage, when a flower is still closed, tube petals produce volatile compounds, and these volatile compounds accumulate in a stigma. And it makes some sense. I really don't know why it's like this, but most likely plant needs this communication to coordinate stigma growth and petal growth. And plus, I think it's really very useful because if you look on petunia stigma, it has a lot of acyl sugars and it's moisture, and bacteria will grow very, very nicely there. But you need to have really healthy stigma for reproduction. And if you sterilize stigma like this, when flowers is opens, it's practically sterile. It will be good for 2 days for fertilization. And then we decided to downregulate actually expression of this gene to prove that indeed this is occur in petals. And what we did, we downregulated expression of TPS1. We see that tubes did not produce these volatile compounds. And here you can see germacrin D, which was very good actually marker for it because germacrin D was produced in, oh, I'm sorry. No, here it's not shown. Yeah. geraniol was produced, in pistils. Geraniol was really very good market here. But you can see these compounds are not produced in tubes. It's not the accumulation in pistils. Now, what we develop also, we develop this complementation assay via gas phase. What we did, we put stigma in magenta box, and then we put tube from another flowers. Practically, we took transgenic stigma and we put wild type tube. And actually, this approach gives us very good tool to study different, genetic backgrounds. And you can see that right away, we were able to restore volatile compounds in a stigma. What was very nice, unexpected, but these transgenic plants have much smaller, stigma size and pistil size, diameter, and it could be really recovered if we would put these transgenic plants, transgenic stigma in wild type tube. It means that gas complementation restores stigma development, and practically pistil size phenotype is independent of pistil genotype, but depends on the tube genotype. But when I saw this, I said, bingo, we have a marker for interaction. Let's try to figure it out. What is the mechanism of this interorgan interaction? And actually, this also shows that stigma development at seed yield depends on terpenoids, which was hormone-like function of this compound previously unknown. And what we did, we generated RNA synthetic sets in day -1 and day 1. And we look for upregulated and downregulated genes. And you can see that it's no practically difference in my day -1, but significant difference was in day 1. And when we did gene ontology term analysis, we found that 35% of term were associated with multiple stress responses, including response to carricine, as well as signal transduction and signal and cell communication. And what are carricines? Carricines are biologically active compound of smoke. Plants don't produce carricines. And carricines are very similar to strigalactones structurally because they have this butanoyl moiety, which is 4-carbon heterocyclic ring. And they actually, although they have similar structures, they induce different responses with some overlap. And although they have also different origin, they actually signal through similar pathways. And upon binding of strigalactone or carricine with their respective receptor, D14 for strigalactone and Chi2 for carricines, receptor is going through conformational changes and interact with MAX2 box complex of E3 ubiquitin ligase system, which facilitates ubiquitination of transcriptional co-suppressors and their proteasomal degradation. And the degradation of transcriptional receptors leads to activation of downstream response. And when we look on our, and one more thing here, carricine responsive genes were found in very widely distributed in plant kingdom and even in plants which grow in non-fire-prone environment. They were very conservative between angiosperm species and they found before land plants, even in xerophytes. This also suggested that probably the function of carricine signaling pathway should be to identify, to respond to unknown endogenous plant signal molecule, which called carricine ligand, which was not known before. However, normally plants have 1 or 2 genes for carricine receptors, and they all belong to conservative clade of receptors. However, 15% of flowering plants in lemons, including Solanaceae plants, they have 4 receptors 2 which belong to intermediate clade and 2 to conservative clade. And petunia indeed has 4 receptors, 2 in intermediate clade and 2 in conserved clade. And here you can see Arabidopsis K2 receptor. And when we check our RNA-seq data set, one of these intermediate receptor expression of one gene responsible for this intermediate receptor was upregulated. When we look on expression of all these 4 genes, which share about 70% identity, we found that K2I and K2B intermediate clades, they have slightly different expression profile, and CA and CB also. But what is interesting, if you look like this, they have similar expression profile. And on chromosome, IA and CB, they clusters together, they're adjacent to each other. And most likely it was like duplication of this cluster and led to this cluster, which also close to each other. One gene, which is IA, was upregulated. And when we compare expression in a stigma, it has the highest expression, the stigma out of the form. What is interesting is that expression of this K2I was negatively correlated with the levels of emitted terpenoids. And you can see here, as soon as there was no terpenoids here, this is TPS tube, which doesn't produce volatiles. But here you can see, again, TPS tube, and you have wild type stigma. The expression of this gene was upregulated. The expression of all other 3 K2s was not changed. We generated these deaf receivers with downregulated K2I, and what we found right away is that stigma was really reduced in these plants. And what it means that most likely carrikin-like signaling pathway is involved in terpene-dependent stigma development. And also what it shows that other K2Is are unable to compensate for reduced K2I activity, likely due to different ligand boundary activity. What we wanted to know if how specific this response is, and we use like different tubes here, carrikins, and we also use plus germacrin, because petunia produced minus germacrin, and you can see that only minus germacrin was able to sustain normal growth of wild-type pistil. And you can see here the same, that minus germacrin was not able even sustain growth of K2I pistils because they were unable to perceive the signal due to downregulation in receptor. When we check different compounds, including caryophyllene, which is saskaterpene, farnesol, saskaterpene, nerolidol, linalool, monoterpene, and eugenol, which is phenylpropane compound, none of them was able to complement or sustain normal growth of tubes. It means that K2I was unable to perceive minus germacrin, and perception of minus germacrin occurs via K2I receptor, and other K2Is have different ligand specificity. We needed to prove really, it's again like a biochemistry, that indeed this receptor perceives this minus germacrin. And what we decided to use MMS. And the way how this works, you take your, we use recombinant protein here, and we incubated this recombinant protein without molecule and with, in the presence of minus germacrin, and then subject for very short protease treatment with protease K for 5 minutes. And then we did trypsin digestion and analyzed protein. And if molecule binds or protects this proteolytic site, then you see changes in protein peptides which you will obtain. And indeed, you can see here that only minus germacrin changed the profile where it was no changes with plus germacrin. And here we use DAR24 as a control. And what is interesting is that we got 303 proteins total we identify, and we had complete coverage. And actually, these peptides were localized around binding site of compound. And then we decided to check if it's really uses, like hurricane signaling pathway. And what is interesting, we developed this transient expression in stigma, which I just said at lab meeting, I said, let's try. And they try and brought me results. And you can see here the practical interaction of Ki2I with XMAX protein. This is pull down assays occurs only in the presence of minus germacline here. And minus germacline is not present, it's not down. And then actually the SMAX is for suppressor, it's greater. And you can see here, we overexpress its max in different backgrounds, wild type, TPS1, which is does not put TPS1 down-regulated plants, which don't produce minus germacline, and Ki2I, which cannot perceive minus germacline. And you can see degradation occurs only in wild type when minus germacline is. And to summarize, perception of volatiles is compound specific, and Ki2I and hurricane sensitive receptor of unique intermediate clade specifically recognizes minus germacline, and minus germacline mediates communication, relies on Ki2I dependent signaling pathway, and shares some transcriptional gene targets with hurricane responses. Ki2I dependent minus germacline signaling transduction operates via MAX2 ubiquitin ligase degradation of MAX1, and practically inability of plants to perceive this minus germacline decreases seeds production. You can see it here. Thus affecting plant fitness. And with this, I want to thank all people who participated in this work from my lab and my collaborators at Purdue University, and John Morgan specifically. It's long-term collaborations here. And on ABC transported with Mark Baldry and the last project on Ki2I, we collaborate with UC Davis and Nitsan Shibekpa. And of course, I want to thank all agencies who provide money, and especially the National Science Foundation, who funded my work for all these years. And with that, I want to thank you, and I would be happy to answer questions.
Very much, Natalia, for this really insightful presentation of all kinds of mechanisms that people really were speculating about for a long time. Other questions, I would like to encourage questions by our students here in the audience, if you have some. Maybe you can start it. Something that's not clear, or any question that comes to mind you can ask.
Yeah, please.
Thank you so much for the great talk. I was curious if you think that any other aspect of global warming might affect cuticle size, which will then affect the volatile compound sort of pathway, if there might be a correlation there or not?
Cuticle thickness, I don't see direct effect of global warming on cuticle thickness, unless it will become even thicker, because, to prevent evaporation of water, right? We wanted to overexpress and get a cuticle thicker to see what will be. So far, I think we did not get clear results, and somehow this project was behind. But it would be really very interesting, aspects to look. What we're looking right now, how wind, will affect emission, because, it's still gradient, and we never excluded diffusion out of the cell, right? And gradient could be also driving force of emission. And as soon as you, eliminate these compounds nearby, probably you can increase emission. But this will be the next step, which we will announce. Yeah. Good question. Sure, I need to look for our results. Back.
So do cell wall features play like a negligible role in the emission of the option compounds then?
It's a barrier, and this is aqueous phase. And because they're lipophilic compounds, they're not soluble. In this case, they're passing, but they always would preferentially partition in membrane bed or in a cuticle. It depends on how far you are and what gradient you have. In this case, lipid transfer proteins, they're just taking and moving them. I have, I have another hypothesis, but I never check it. I just need hands for it. But I saw one paper in E. coli system where interaction of two proteins change specificity of transporter. And because it's so many different compounds are coming out, right? You can have transporter for each of them, but it should be some kind of specificity, right? Of course, it's binding constant, which is different from different compounds which drive it. But also, I thought maybe lipid transfer protein can interact somehow with ABC transporter on a side, and then maybe preferentially move one of the other compound. But in general, this is a barrier, and it's really liquid barrier across on emission path. Yeah.
Variants presentation. So I just want to know, what's your next step in this research? Yeah.
Okay. First of all, we want to know the mechanism more about this K2I, perception, and maybe some connection with ethylene, what we saw. What we don't know, what is the function of the other three transporters, and we are generating right now transgenic plants for the other three transporters, because intermediate clade nobody knew, and the function is not known. What is really very surprising for me, such serious specificity for one compound, and why it's minus germacrin. I could not find anything in the literature, but what I found that MOS actually has a specific receptor for minus germacrin specifically. Why? I don't know. Yeah, and it will be interesting to see how broadly, this function distributed. This is why we also use tomatoes to check function. Yeah.
That was an incredible talk, so thank you so much. You showed that receptor kind of communication in the flower. Do you think something like that could exist in leaf tissue, another part of the plant?
For signal transduction, most likely, but initially what I was thinking is that concentration in the air, right, it's relatively low because it's diluted, right? And now you have a leaf. Do we need any concentration mechanism for signal transduction? The way how volatiles can get inside the cell in two ways, through stomata or through cuticle, right? The involvement of stomata is not really clearly shown. This community is still debating, If they're debating between themselves, I don't want to stay on a line because they know much more than I do know in this area, right? But what I want to say is that when we calculate it for several compounds, really you don't need any concentration mechanism. It will be enough to go through stomata, But here you need to consider that stomata close during night. And in this case, if the signal is coming through night, you will not get this signal and it will be perceived, right? Much longer, you will have like response through cuticle. Cuticle can accumulate and you can respond it much longer. Which pathway and how it works, I really don't know. But in the beginning, I need to know at least from people in ecology what compound I'm using to treat, and I could not get this information from them, unfortunately.
Wonderful talk. Nothing like seeing a good hypothesis be disproven, you know, in terms of the thickness of the cuticle correlated emission. You know, you elegantly showed that the metabolism of the intermediates is going down, and that's accounting for less. How is the internal part of the cell able to sense a thinner cuticle?
The way how it works, you know, you stop to accumulate compounds inside the cuticle. They build up inside cell. And when they build up, probably there is a sensing mechanism. I was thinking about it, and right now I have two hypotheses, how by small molecule you can regulate expression of genes, because here you have a small molecule, here you have a gene. It's two different worlds, right? And one hypothesis is that transcription factor or repressor will bind with a small molecule. However, nobody showed any binding site between all these transcription factors for small molecules, right? Another hypothesis, and probably this is what is going on, you have biosynthetic gene releasing volatiles. When amount is accumulates, probably they stop to release. And I think what happens, they probably change their subcellular localization, go on the nucleus, and interact with transcription factors. And this is what we are changing. And actually what we did turbo ID, and we identified one already biosynthetic enzyme, which was together with, you know, like acetylase and deacetylase, which acetylase, you know, change transcription. I don't know, future will show.
All right. Well, one last question. Yeah.
No, two. We need from students, you know.
So I'm just curious. So you said you were part of the the paradigm shift when people didn't want to believe that emission was different than just diffusion. Yeah, was that discouraging to you when, you know, most of the scientific community was against that change? And do you feel like there's any other status quo things that are like ready to be overthrown? Like, is there anything that the scientific community?
You know, it's very difficult to say, but what I want to say, it's not easy to change paradigms. You know, people don't believe it. It's already set. And even my friend said, stop it. It's diffusion, you know. But you do see what I want to say. Be focused and see all these small inconsistencies. These small inconsistencies will tell you a lot. And I started to see, for example, in old flowers, I have huge internal pool and no emission. It was in Snapdragon. How in the world, if it's diffusion, what is keeping it inside? You know, this is why I truly believe in it, that it should be something there. But it took a lot of years to prove it, you know, and a lot of thinking how it works. Yeah.
Okay, one last question.
I know you mentioned Fick's first law, which relates like the flux and the diffusion of like the volatiles to the cuticle. My question is like, why is this flux like relevant? Like, what studies are scientists doing?
Well, this is normally what physicists and, you know, engineers use to calculate the flux. You know, it's normal formula. We did not invent anything here.
Just wondering like why it would be helpful.
Why it was helpful? Because we wanted to calculate how much we should have in order to sustain it by diffusion alone. Because we were able to measure two parameters. And now we just model it as a diffusion. And when we model as a diffusion, how much should be inside? This is only why we use this calculator. And when we saw this millimolar range, it was crystal clear that it's not possible to sustain it, you know, like to sustain healthy cell with such concentration of volatiles inside.
All right, well, thank you very much for your great questions, and let's thank Natalia one more time.